Good morning, everybody. My name is Adam Boyd. I'm from the Department of Energy's National Nuclear Security Administration, and I'll be moderating this morning's session. This morning's invited talk is Dr. Rick Stevens. Rick Stevens is a University of Chicago professor and Associate Laboratory Director for the Computing, Environment, and Life Sciences Directorate at Argonne National Laboratory. Among his many projects, Dr. Stevens co-leads the DOE National Laboratory Group developing the Exascale Computing Initiative. He is the principal investigator for the Patrick Bioinformatics Resource Center, which develops comparative analysis tools for infectious disease research. He also is co-PI for the Systems Biology Knowledge Base, or KBase, which is building a comprehensive computing environment for biological discovery.
Thanks a lot, and thanks for inviting me. And this room is too big. Gosh, it's like an interesting distribution of people. You know, it's sparse up here and gets denser in the back. So I'll have to walk in the middle, but I can do that with this clicker. So I was asked to talk about two initiatives that are happening in the U.S. government that affect DOE. And both involve partnerships with other agencies. So I'm going to talk about the Cancer Moonshot initiative, and I'm going to talk about the BRAIN program. And this is kind of a high-speed drive-by. There's probably too much stuff in here, but I thought I'd kind of give you a flavor of these projects. Now, some of you might be wondering, how do you make the connection between cancer and brain and DOE, right? And I'll try to make that clear, but one of the things that may not be totally obvious for people that are just coming into the research community is that DOE has an unprecedented concentration of scientific computing compared to other institutions in the country. And if you look at NIH and you say, well, how many supercomputers does NIH have? Anybody know? One, maybe, if you counted it. The U.S. government research agencies are quite distinct in terms of the portfolio of capabilities that they have. And that's a challenge if you want to bring a multidisciplinary approach to attacking problems that are not in, say, the mission space of one agency with the resources or the capabilities of another agency. So this has been widely recognized by OSTP and by the agencies themselves, but we have the scientific landscape and funding agency landscape in this country as a result of a large number of kind of accidents in some sense as to how the agencies have been organized. And so this is an effort to kind of span multiple agencies to bring together the types of resources on the problems that we actually need. So because I probably won't finish everything that I want to talk about up front, I just want to acknowledge that there's content in here from Argonne, Berkeley, Oak Ridge, Livermore, Los Alamos, National Cancer Institute, UCSF, Harvard, Chicago, and here's people that have either sent me stuff or that we're collaborating with on this project. So earlier this year, there was an announcement in Physics Today or something that had this little article that said cancer, brain research, and supercomputing. And what this article tried to do, see if this pointer works, yeah, was to argue that there's this National Strategic Computing Initiative, which is an umbrella of many agencies under which the Exascale program that DOE is working on is under, including other advanced computing R&D topics. And inside that framework, there's a notion that DOE will provide capabilities to other agencies that need supercomputing, applied math, modeling, and simulation for their own initiatives. And there's two other initiatives that were launched over the last couple of years, one in precision medicine, which includes cancer as one of its targets, and the BRAIN initiative. And so I'll talk a little bit about the cancer initiative and the projects we're doing with DOE, and then I'll do some on the BRAIN. So earlier this spring, there was I guess January, there was this announcement. Joe Biden, who had lost his son to brain cancer, was asked or pushed by the president to coordinate a big national all-government approach to pushing forward on precision medicine with a particular focus on cancer, and this was labeled the cancer moonshot, right? And moonshot is a phrase that lots of people use, trying to capture the mythical notion that the whole government's working to achieve some, goal, like, putting somebody on the moon. Of course, without all of the actual logic behind something like a real moonshot. But anyway, think of this as a way to accelerate research in cancer and to pioneer this notion of predictive medicine and doing that in cancer, but then apply it to other areas as well. And just to give you a 2-second tutorial on cancer, right? So we have, you all have lots of cells, and the cells are normally, after you develop, like as an adult, I guess, which we're, well, not maybe some of us aren't really adults yet, but your cells probably think you're an adult. They mostly stop growing, and they're in a kind of a quasi-homeostatic state that in this diagram is labeled G0, kind of a happy, not dividing, just doing their normal function state. They're fully differentiated. But the cell, of course, has machinery in here to trigger itself to divide and to grow, and this was, of course, essential when the organism was being developed. And one set of classes of cancers happen when the cell flips back into this growth mode when it shouldn't be, right? And I'll give you a little sense of how this works. So one of the ways to trigger growth stimulus is through a set of molecular signals that bind to these receptors that are in the cell membrane. And there's several different growth factors that do this. They bind to this receptor. It causes a set of chemical transformations on the molecules, which sets into motion a cascade, what's called a signaling cascade. So a series of molecules interacting with other molecules. In this case, this kinase receptor gets activated. It's kind of like a Rube Goldberg machine in some sense. There's balls running down here, and we get an adapter protein that then activates a thing called a RAS activation protein. RAS is a little protein that hangs out in the membrane. Each of your cells have many copies of this. And normally it's turned off, and when it gets turned on, it starts propagating more signals to cause the cell to divide. And there's this cascade here, this ERK cascade that goes from the RAS molecule to the RAF molecule to MEK, ERK, and so forth. And then a whole bunch of cellular machinery gets turned on that causes the cell eventually to start dividing. Okay? And if I put this in a networky kind of diagram, because we're all computing people, you can kind of get this. Up on the top here, not really meant to read this, but just appreciate there's a lot of complexity here, okay? There's molecular, and over the last 50 years of studying cancer and cancer biology, we've deciphered a lot of these pathways, and this upper part here is the basically the RAS signaling pathway. Here's this RAS molecule in the middle. So these EGRF receptors here trigger this guy triggers a bunch of stuff, eventually goes down here and causes the cell to proliferate. This set of pathways down here cause the cell to kill itself, and so this is a kind of a balance that that cells have to undergo, and cancer occurs when these two things become out of balance, when growth is overcoming the signals to commit suicide if something is wrong. Okay, so you think of this set of network pathways. Now, this is kind of just a background. So NCI has been attacking cancer problems for a long time. We've had the war on cancer. And cancer survival rates are improving, and I'll have some slides on that a little bit later. But one of the key challenges is that NCI has not brought the full capability of advanced modeling and simulation and machine learning to these problems in cancer. So about 2 years ago, there started to be a number of conversations between DOE lab researchers and the National Cancer Institute to try to come up with a set of concepts of what we could work on that would try to bring these two things together. And that eventually got labeled this horrible name, Joint Design of Advanced Computing Solutions for Cancer, which you can't actually, it's an acronym that you can't pronounce and you can't even remember it. But basically it's bringing computing and modeling to cancer and bringing the problems from cancer to driving requirements in computing, including future computer architectures. And we basically chose 3 problems. So the first one is a problem out of cancer biology that I kind of motivated earlier, which is to really get a deep understanding of how these RAS pathways work. And this is essentially simulating the molecular interactions in these pathways and to look for potential new drug targets, molecules that we can inhibit with, say, small molecules or some other mechanism that would result in, like, throwing a wrench into the gears to stop the bad things happening in these cancer cells. And I'll talk about this one a little bit more. The second one is to bring machine learning and modeling techniques to understand and interpret preclinical models. So we can build, we can create tumor-like cells that can grow in a dish, and we can screen these at high throughput with small molecules, and we can also do molecular assays on these cell lines. And we want to be able to project from what we learn in cell lines in a dish into real tumors, and that's essentially goal number 2. And goal number 3 is to make sense out of the cancer surveillance data that we've been collecting across the country to build population scale models. And this also involves machine learning and natural language interpretation, and then ultimately building agent-based models to calculate patient trajectories. And I'll try to give you a sense of what we're doing in each of these 3 areas. So in the RAS example, as I showed on the first diagram, and this kind of looks like a circuit diagram, I figured you might be a little more comfortable with it. So you have these modules. These are, we reverse engineer our understanding of how the cell works, and we label it in these modules. But of course, evolution didn't necessarily build it this way. But we have these modules that are essentially the RAS interactions, and we're trying to essentially have a detailed simulation of the interactions in this component right here to look for new drug targets. And the reason that RAS is a really interesting target is about 30% of cancers actually involve mutations in the RAS protein that cause it to get stuck in this on position. Okay? And some of the worst cancers, the hardest to treat cancers, are in this category. So if we could find a way to stop that interaction, we would have significant progress in approximately 30% of the cancers. And so far, RAS has been resistant to designing drugs that can bind to it because it has a particular shape. And so we're looking sort of downstream in that pathway. So here's sort of what that, if I zoom in on that RAS piece, you can get a sense that there's dozens of pathways, molecular interactions after that, that ultimately result in the cell doing what it's going to do. And each of these potentially becomes a place we could insert an inhibitor to block that pathway. And the key is trying to understand the dynamics of this system, both at the kind of large-scale network level dynamics, but also at the atomic level, so we could design inhibitors. So the way that we're going to do this is by building a molecular dynamics simulation that's capable of reproducing the large-scale molecular interactions of these protein molecules and their signaling components, and be able to zoom in and zoom out based on what we learn, either down to the quantum mechanical level to understand, say, the binding of signaling molecules to the protein, or to zoom out to coarse level to kind of get a sense of the larger scale landscape. And to do this, coupling it with experimental data that's being collected at the National Cancer Institute, and to manage these simulations automatically with essentially unsupervised machine learning that's looking over the state space that the molecular dynamics is running in to choose the next move. So think of this as trying to automate the process of deciding which simulations to run at which resolution to maximize the information content in that pathway. And that's the project. It involves building this kind of zoomable molecular dynamics. It involves scaling up the machine learning to take the state space information from those simulations and to plan next moves, and to also reconstruct, in some sense, the interaction networks and what we know and how much uncertainty we have in those pathways. Okay? So that's the RAS problem. The second problem is the one I'm leading, and this is in this preclinical screening. So one of the problems we have is that over 50% of patients don't respond to chemotherapy for many tumors. And this is a big problem. We know that standard chemotherapy has all kinds of difficulties. Eventually, It has huge toxicity problems for cells that are not the cancer targets. Your cells become resistant to it using all kinds of drug resistance mechanisms that the cells have built in to defend themselves, not that different from how bacteria become resistant to antibiotics. And so one of the more recent strategies has been to try to use molecular assays, that is, say, genomics information or gene expression information, to somehow differentiate those tumors that might be particularly responsive to a given drug and immediately give the patient that drug as opposed to some kind of random, not random, but standardized therapy that's not customized. Another thing is to try to add additional drugs together, whether it's to add different combinations of chemotherapeutic drugs, or to add immunostimulants that would work in conjunction with the chemotherapy to try to invoke the immune system to help out. And this is a huge challenge. And so what we're trying to do is figure out how to actually streamline the building of models that would give us a recommendation for this patient, try these set of drugs in this order or this combination of drugs. So one of the challenges with cancer is that cancer mutations occur. Usually there's a set of driver mutations, a handful. Typically it takes about 3 mutations to result in a normal cell becoming cancerous. And all of us are having cancer cells created all the time in your body. Most of the time your immune system takes care of them before you even worry about it. Over your lifetime, over our lifetime, about 40% of us will actually get cancer. And many of us, maybe a third of those, will actually die of that cancer. So this is a ubiquitous disease. And as we extend our lifetimes, that fraction is actually probably going to go up. That is, if we were going to live to be 150 years old, we would have that say, between 90 and 50 years, another 60 years worth of life, we would be subject to a huge amount of cancers occurring. So one of the reasons we want to push forward on this is partially to drop that burden down, but also to make it possible to actually live longer and healthier lives. Well, so we go back to a cancer. Here's a snapshot of a biopsy of a tumor, and just to take away from this, there's about 200 little dots here. Each one of these were sequenced. Okay? And the question that was being asked here is, how similar are the cells in that spot? This whole thing across here is about 1 centimeter. Okay? How similar are these cells to each other from the standpoint of the mutations that they're carrying? Traditional theory would say that you have a set of driver mutations, a handful, you pick up some passenger mutations along the way, and Darwinian evolution is kind of applying, and so the tumor would be somewhat homogeneous in terms of these mutations. It turns out that there were over 100 million distinct mutations in this one tumor. And the evidence, I think I got a shot here, this is a kind of a lineage of how the cells are related to each other. And what this showed is that statistically, there's way more mutations than Darwinian evolution would actually explain. And so we now believe that cancer cells in fact, are not subject to Darwinian evolution, at least initially. They're subject to kind of what we call neutral selection, which means they're just kind of growing, accumulating mutations. And this accounts for why tumors are so resistant to drugs, because they're differentiating constantly. And you might hit this tumor here with a drug that would kill 90% of these types, but 10% would still be resistant. You'd kill all those guys, and that 10% now would grow back up. And this is kind of the fundamental problem. So trying to tune the drug to the cancer is this overarching challenge. I think I already said that. So one of the projects that the NCI is doing is trying to screen combinations of molecules. So there's about 100 small molecules that are FDA approved for oncology treatment. And recently they started an experimental screen that's basically screening all pairs of these 100 molecules. So it's about 5,000 drug pairs, and they have about 60 different cell lines. So this is about 300,000 experiments, and we triplicate it or more than that to get the different dosing levels. And one of the output, you can't really read this, but it turns out about 1% of those combinations are unreasonably effective. That is, the two compounds together are super lethal to the cells. Here's just a chart here. This is what happens if you, the null case, or black line's the null case, this pink line here are two compounds that just kind of together are synergetic and really kill the organism. One thing we're trying to understand is, can we predict these combinations? The reason we want to do that is the actual library of compounds that have been screened on tissue over the last 30 years is about 500,000 compounds. So just taking pairs of 100 results in 4 million screens. We can't do all pairs of 500,000. That would take us forever. So we want to apply computational methods to try to predict which pairs would actually be effective. Not only that, we want to actually do this in things that are more related to actual patient tumors. So there's this concept of patient-derived xenograft models. So xenograft models here means that the tissue comes from one type of organism and is planted in another organism. In this case, the tissue is a tumor resected from a patient, and it's implanted subcutaneously in a mouse, an immune-suppressed mouse, so you can grow up a human tumor inside the mouse, and that's a much more realistic environment for testing drugs than human cell lines that have been growing in a dish. And so one of the challenges that we have is, can we take the results that work in a dish and project them onto the mouse, and can we take the mouse and project it into the patient? And that's kind of the trajectory. So the way this works is we've been building machine learning-based predictive models. These are not mechanistic. They don't explain why the prediction works. They're just giving us predictions, and in some cases they can be quite accurate. We're trying to then wrap uncertainty and optimal experimental design around the output of these models to say, look, here's additional experiments you should do to improve the accuracy of these predictive models. And then finally, we want to mine these machine learning models to pull out hypotheses that we can go in and test to understand mechanism, and ultimately combine mechanistic models with these machine learning models. I was chatting last night with some folks that are doing deep learning on some problems in plasma physics, and here's a kind of formulation of the deep learning version of this cancer problem, where we have descriptions of the drugs, the drug target, molecular fingerprints, and so forth, various descriptors as a set of features for the drug, and we have a lot of features that characterize the cell line, and what we're trying to predict in this case is the drug response. And if you kind of drew a diagram, it would look something like this: molecular characteristics of the tumor and drug information, and this is just a schematic here, it's not the actual networks, but we're trying to learn the relationship between the tumor types, the specifics, and the drugs to be able to predict what that drug response would be, or alternatively, try to predict the relative order you should give drugs. Okay. And I think I'll skip that. So the final project here is this cancer surveillance system. So in the US, we have a national law that says if you have cancer, your information will go into a national repository. So we have 100% kind of surveillance cover in the US, and this was done years ago to try to understand, the cancer demographics, and it's very similar in Europe. Third world doesn't have this so much, developing world. But because we have this information, we know a lot about the population level statistics in cancer. So for example, this is survival rates. This is the number of years on average that you survive after a cancer diagnosis in the US versus Europe. It's a couple years extra. Here's the average cost for cancer therapy in the US versus Europe, right? So you can more or less say if you pay more, you get a little bit more. Here's kind of high-level statistics. So in the US right now, you have about a 67% survival rate for 5 years after a diagnosis. But the number of cases, we get about 1.7 million cases a year, about 600,000 people die per year. This is the number I was quoting earlier, that about 40% of men and women will be diagnosed with cancer at some time in their lifetime, right? And so we can get all this from the global data. And what we are trying to collect in this global data is everything from the pathology reports, the molecular details of the cancer, if there was radiation treatment, what was the initial treatment, how effective that was there a secondary treatment, what was the survival and the long-term outcome. And build this across the entire population, then be able to extract information from this database and start to build models that would allow us to experiment with kind of high-level policy issues about what types of treatment should be prioritized in different kinds of cancer in different parts of the country. So again, this is the third pilot, and at the heart of this problem is interpreting pathology reports automatically. These are text reports that are written in a very specialized language that defy, automated dumping into the databases, but we need to be able to do that in order to be able to build, say, agent-based models that would allow us to compute on this data. So there's a big natural language problem here and a kind of data analytic machine learning to extract information and then try to make sense out of it. Again, apply uncertainty quantification, key mathematical ideas to understand where do we have the biggest gaps in our knowledge and what would be experimental strategies or better surveillance strategies that would reduce the uncertainty in our understanding. And then ultimately building models that would then integrate across the pilot 2 information about, say, the precision oncology treatment and all the way back to the molecular mechanisms. We want to go from the mechanism all the way to the population. So that's kind of the cancer pilot. And this involves 4 national labs, a number of universities, Harvard and Chicago and others, and some pharmaceutical companies and partnerships with IBM and Intel, and of course, the primary high-level partnership between NIH and the Department of Energy to tie this stuff together. So this is a kind of unique arrangement. We're kind of in the middle of our first year of getting this going, and I think this is a model for the future of how NIH and DOE will work together, bringing the mathematics, the supercomputing, and the machine learning together. And what we imagine to come out of this that will affect DOE more generally is the following. So, historically, we've built large-scale machines to support, numerical simulation, right? PDEs, fundamentally. So we have machines that have pretty good architectural characteristics for doing that. They have reasonable memory configurations. They have tight coupling in the interconnects. They have strong I/O systems and so on, but they've been kind of optimized over decades for numerical PDEs, the first order. But what we're seeing that the broader science needs have to bring in scalable data analytics for data-centric problems, and we're seeing more and more use of machine learning, particularly deep learning, as challenging this. And so for the future, we want our supercomputers to actually not be optimized just for this part of the problem, but to be able to cope with the entire set of the problem. So this is something we expect to learn from projects like this cancer project. Another spin-off is that the labs are collaborating together to try to build a machine learning framework that is actually scalable on the platforms. And in the case of the cancer projects, we've got these 3 different projects, but they all have machine learning at the heart of it. And this is a scope of a project that we're booting up to optimize machine learning for these big machines. So that's the cancer scenario. Let me try to jump on the brain for a second. So in addition to cancer problems, the government has been working on a high-level initiative to push forward on the brain and neuroscience, and the shorthand for that is called the Brain Initiative. I'll explain that in a second. But just to give you some basic concept here, the brain has about 100 billion neurons and quite a bit more than 100 trillion synapses, but this is kind of a good rough estimate. If you were trying to put this in perspective, the amount of information to characterize that at the current way in which we do imaging would be about a zettabyte. So this is not going to happen anytime soon. This volume of data is the equivalent of if we were capturing the entire internet traffic for a whole year in one place, right? So this is kind of the magnitude of the challenge if we wanted to reconstruct the brain at the level of the deep synaptic networks. But I'll talk about how we're doing that. So in 2013, Obama announced this initiative called the BRAIN. Stands for Brain Research Through Advanced Innovative Neurotechnologies. Okay, it's one of these reverse acronyms. As you choose the acronym, you try to find the title that matches it. This has been cooking for quite a while. This is the window here. The big picture here is fundamentally we want to map the circuits of the brain. So a static map of the connections. Of course, each brain is different. Everybody's a snowflake, if you want to think of it that way. There are millions of snowflakes. So we want to map at least one brain, but ultimately we want to understand how to map brains in general statically. Then we also want to understand how does the electrical and chemical behavior in this brain fluctuate over time. There's a functional or a dynamic map of the brain. How does that couple with specific circuits? How does that interplay actually create cognition and behavior, but also how does it result in disease? Okay? And the approach here is to do this not just for humans, but to do it across simpler nervous systems. That is, we want to go from nervous systems, from invertebrates all the way up the phylogenetic tree so that we gain a kind of comprehensive understanding of how nervous systems work and can understand the human in the context of that evolutionary background. One of the kind of selling points of this initiative is they tried to model it after the Human Genome Program. Okay, so the Genome Program took about 10 years, took about $3.5 billion in the US, more internationally, and has led to a kind of a revolution in some sense of the way we think about medicine, right? And so the idea here is, could we do something similar by a deep dive in the brain that would have lots of spin-offs, and in the process would help us understand and attack diseases like Alzheimer's and Parkinson's and PTSD and traumatic brain injury and so on. So that's the motivation. If you're on the Hill, if you're on Congress, and you're talking to them, and I know we got this award about how to do, technical, how to explain science, to non-technical audiences, this is the argument for doing this, right? We're never going to really understand these diseases unless we actually understand the underlying organ in which these diseases occur. The other argument is an economic one. So the Genome Program invested almost $4 billion, and based on some accounting, right, since that investment has generated an economic lift of about $800 billion, or about $140 per dollar as an ROI. So this is a huge, return on investment if you could actually make this happen. So could we do the same thing in neuroscience, right? So I already went through the high-level goal, but to do this, we have to actually understand how many different cell types are in the brain, how are they wired up, how do they work dynamically, how do they play together to create behavior, what are the kind of fundamental principles of the architecture, how do brains really work, apply this to human neuroscience, and apply this human neuroscience to medicine and translational goals like disease. So this is what they're trying to do. It's got a lofty target, kind of a 10-year objective. There's a beautiful document. If you ever want to see I think a kind of a high exemplar of how to do a strategic planning document for a science initiative, this is a good document. It has some flaws, but it's pretty good. It lays out the vision. If you just Google, you'll find it. The research, how they're going to do it, what the costs look like. It was the product of about 6 workshops and about 100 people. One of the flaws from my perspective is that it didn't include enough math and computer science people. So you guys, as the next generation, if you ever hear there's going to be some project, like this happening, make sure you tell your colleagues to invite math and computer science people, because out of this entire report of 146 pages, computer science is mentioned 5 times, and math is mentioned 4 times. And this idea that somehow you're going to acquire all this data and make sense out of it without any math or computer science is, of course, a little bit odd, to say the least, right? But in hindsight, the DOE is coming in to participate here. Here's kind of what these initiatives look like. This is kind of a generic chart for funding ramps and initiatives, right? So initiatives always have some beginning, and they have some budget ramp, and then kind of a plateau, and then they kind of roll over. And in this case, this is how do you spend billions of dollars, and you do it this way. You have some infrastructure, you've got some technology, and you have some science, right? And these things always kind of flatten out with some permanent budget lift for the fields. And you can ask Barb, does DOE plan initiatives like this? Yes, they always look like this. Of course, it's been up and running for a couple of years, and it funds lots of people at lots of places. This kind of just follows the population distribution, so nothing really amazing there. In FY16, here's roughly the budgets. These two didn't have their budgets public, but basically a couple $100 million. NIH spending the majority of it. DARPA, I'll have another slide on this, what they're doing in NSF. And more recently in FY17, DOE is getting into this with some initial budget requests. So at DARPA, as you might imagine, their investments in neuroscience are really aimed at a couple of goals. One, primarily rehabilitation of war fighters, right? So people that got injured in war, they've lost limbs or lost cognitive functions or whatever, and they want to restore this. So whether it's restoring memories, revolutionizing prosthetics, having neural implants that can, bridge damage to the spinal cord or whatever. So there's a lot of work really in this kind of treatment of warfighters. That's kind of the DARPA focus. The NSF focus is very broad. It's kind of multi-scale integration of activities. It's kind of neurotechnology, very things like Brainbow. That's where you go in and you modify the proteins in neural cells to have a rainbow of fluorescent dyes, so then you can hit them with lasers and they kind of light up in different colors, and so you can start to see the structure. Quantitative and modeling, kind of technologies for sensing, and of course workforce development, training people in neuroscience. So the reason the brain is a real challenge is that it has a lot of scales, right? So your brain, it's roughly about 1 liter in volume, and you have folds at a kind of a centimeter scale. You've got some large-scale neural structures on the order of 1 millimeter. You've got dendritic spines and so forth, all the way down to synapse details that are kind of submicron, kind of at the 10 nanometer scale. And one of the fundamental challenges is building this underlying map. Okay, and this is the kind of canonical workflow in doing that. So we typically will do this in mice, but you can do it in other animals too. So you would extract a mouse brain, you would section this brain using, I'll show you a picture of the sectioner, into very thin slices, kind of 30 nanometer thick slices. You use a microscope. In our case, it's a multi-beam scanning electron microscope. You get thousands of image slices. Then you have to align these and line them up in registration-wise around fiducials or patterns that you discern. And then you want to segment this, which is find the parts in each image that align to make overall structures, and then do analysis on the resulting derived structures. So this is a fast guide through this. So in order to slice a brain into 30 nanometer slices, you have to have a diamond knife that does this. This thing is cryo-cooled. It slices off a thin layer. There's a liquid bath here. It floats onto a piece of Kapton tape. Kapton tape was invented, by the way, for the Moonshot, the original Moonshot. It was what they insulated the lunar excursion module in, just as a piece of historical data. This Kapton tape flows on a little assembly line here, and that's what these things look like. It deposits a little slice of the brain onto this tape, and then these tapes get mounted into a round, literally on a silicon wafer, that then is sputter-coated with carbon, so we can then image this. They go into a microscope. Here's the outside of a picture. This is Jeff Lichtman's lab up at Harvard, who's kind of the godfather of doing this. guys from Zeiss, Hans-Peter Pfister from Harvard has been working on some of the imaging, other colleagues. But behind them is this microscope. And this microscope has 61 parallel beams, and it produces, well, never mind. We're working on trying to scale this up, but some of the data volumes here are pretty impressive, petabyte per day. We do that. But here's what comes out of that microscope. So it's a tiled image. Each one of these little squares is coming from one of the beams, and we have to stitch them together initially, and then make this stack. Okay, the stack, each slice is about 30 nanometers. For this first experiment, we have about 2,000 slices. This represents the idea of how do you do segmentation. So on the right here, for every one of these slices, a human went in here and clicked and gave a label to each one of these objects and said it is a projection of the object from the image below, and so forth. You stack these up. That was used as training data for a neural network that then was able to apply to automatically segment, and it gets a lot of it right. It still gets some things wrong, but this is an open area of research. It's a huge problem. The test case that was done over the last 2 years is a really tiny volume. It's about a 600,000th of a cubic millimeter. Here's kind of a movie. So why these things are cylindrical, they were punched using a biopsy punch. This was the imaging, this was the reconstruction and the identification of objects. This was just pulled apart graphically so you can see what's going on there. There's a spiny dendrite in the middle. The second here will pull it apart and the different cell types. Remember that one of the fundamental things in the initiative is understanding the different types of cells that are in a brain. So here you've got the helper cells in the middle, astrocytes. You've got axon structures here and dendrite structures, and there's right now the count on the number of cells cell types is approaching hundreds. See if I can get this to advance. Hello? Okay. We're also trying to do this with X-rays. At Argonne, we have the advanced photon source, one of the world's brightest X-ray sources. And it'd be nice to be able to do this without actually having to do the slices, or to do the X-rays and then slice. And X-rays currently don't have the same resolution as the electron microscope, but they do produce a huge amount of data. This is a tomographic imaging moving through through a punch slice. So you're seeing the neural bodies here and vasculature as well. And so what we hope to do is, of course, increase the resolution of the X-rays and use this data as kind of the rough guide in which to guide the electron microscope. Let me try to, from that, you can get reconstructions that look like this, that are just purely from the X-ray data without actually having to do the slices. Once you have these objects, you then want to do a reverse engineering algorithm to try to recover the wiring diagram. And here's an example of a database that's done that. So each of these entries in the database is a neuron, and it contains the architecture of that neuron and who it's connected to. So this is the reverse engineered network. And the idea is that once you reverse engineer these networks, you can then derive some kind of a brain circuit, and then you can do modeling on that circuit. Okay? So, yeah, I'm just skip through this. So looping this back to DOE, Oscar is well positioned to contribute to this because we have people that can do theory and modeling. We have all kinds of techniques for analyzing and acquiring dynamic data, and also map that onto the static structure. So this is a grand challenge that's going to be around for a long time. It's something maybe some of you will get involved with. So with that, I will say thanks, and I'm happy to answer any questions if we have any time. So thank you.